Hi, my name is Gabriel, and in this video, I'll be presenting the main result of our latest publication, entitled Critical Review of Conformational B-cell epitope prediction methods. But before we dive into the results, I will briefly introduce the topic. Conformational B-cell epitopes are patches of surface residues that are found on proteins, and these patches are bound by specific antibodies. To determine where these patches are located, researchers use experimental techniques such as X-ray crystallography or mutation experiments. But these methods can be very expensive and time-consuming. To help speed up the epitope discovery process, computational biologists have developed different machine learning models that attempt to predict where these epitope patches are located on a protein. In the past 20 years, many of these models have been published. And so our question is, which one's actually the best? To answer this question, we performed a rigorous and independent benchmarking of 9 popular methods. Briefly, the benchmarking methodology was the following. First, we collected a high-quality and non-redundant dataset of over 250 antigen structures, and for each antigen, we included all of its known conformational B-cell epitopes. Then, to ensure a fair comparison between the 9 methods, each one was evaluated on antigens that were not part of its training dataset. Finally, we only considered the prediction made for surface residues during our evaluation, as it is trivial to predict that residues at the protein core cannot be bound by antibodies, as shown in the picture. The methods that were evaluated include 2 sequence-based methods, 6 structure-based methods, and 1 antibody-specific epitope predictor, EpiPred, which is a new kind of model that uses antibody information for predictions. We calculated the AUC of each method on our benchmark dataset, and here are the results. As you can see, all the methods achieve very limited performances, as they all reach an AUC below 0.6. In fact, in our paper, we show that only 2 of these methods, namely Tiscotope2 and bpro, perform significantly better than randomly predicted surface patches. Finally, we also checked whether combining the predictions of multiple models into a consensus model would give better results. But unfortunately, this approach didn't perform very well either. So, what conclusions can we make from these results? Well, firstly, it is clear that the accuracy of these 9 methods is currently too unreliable to be used in practice, and consensus schemes that combine multiple models do not overcome this problem. Therefore, new methods need to be developed and rigorously validated. It could be interesting for these new methods to try to incorporate new features, such as, for example, the presence of glycosylations, which is known to be very unfavorable for antibody binding. Finally, I would like to leave you with the following thought. What if any protein surface could actually be recognized by one of our antibodies? What if antibodies had precisely evolved to be able to bind almost any surface protein? If this were to be the case, then trying to predict where epitopes are located, independently of antibody information, might actually be futile. For example, there are antigens such as lysozyme that have been extensively characterized, and whose surface is almost completely covered with B-cell epitopes. If you'd like to know more details about this paper, you can find the link in the video description below. Thank you, and see you in our next video.